Record.
Okay, let's go ahead and get ourselves started. Okay, we left off. We're getting ready to talk about the spinal nerves. And so there's a couple of structural things we want to kind of clear out of the way. First of all, it's going to seem a little abstract until we actually see a picture of it. Should be a little more explanatory for that, but that's some structures associated with the spinal nerves. So a couple of things that we refer to as a ramus, basically the dorsal and the ventral ramus. These are connections that lead off of the spinal nerve, and they kind of start to connect to more nerve structures. Now, one of the important things to remember is that essentially what you have is a network of networks. So the nerves are notorious for merging together and then splitting apart and then merging again downstream and then splitting apart and merging. And so they're constantly doing this back and forth. But at the very beginning, we have a couple of these little offshoots, the dorsal and the ventral ramus. The dorsal ramus is basically going to be responsible for hitting those deep muscles of the trunk. So the vertebral column, the skin near the midline of the back. So it kind of wraps around the backside and is kind of going to some of those deeper muscles. The ventral ramus is basically depends on where it's going. So there's the spinal cord itself is sort of split up. Once you start to exit the spinal cord and you move through your roots, your dorsal and ventral roots, then what's going to happen is you're going to start to break up into what's called a plexus, which is essentially a network of nerves. And those plexuses are plexuses, however, whatever the plural is for plexus, basically will move throughout different localities of the body. And so the ventral ramus itself in the thoracic region will hit the intercostal nerves. These obviously are innervating the intercostal muscles, which are in between the ribs, and the skin that's just in that thoracic area. Kind of what we think of as our upper chest, essentially. The rest of it, however, the rami from the spinal nerves, or the roots of the plexus, basically, before it kind of starts to spread out and sort of form these huge networks, will basically feed 5 regional areas. So, for instance, the ventral ramus of C1 to C4 will form what we refer to as the cervical plexus. The ramus that covers C5 to T1 will form what we refer to as the brachial plexus. The ramus that innervates L1 to L4 is going to be forming what we refer to as the lumbar plexus. And then from L4 to S4, it's the sacral plexus, and the S5 is going to be the coccygeal plexus. Now then, also along with the sympathetic nervous system area, we also have what's called communicating rami that basically will connect to a structure called the sympathetic chain ganglia. So it's basically a network of ganglia that forms kind of a parallel chain that runs up and down parallel to the spinal cord. So just to kind of give you a little bit of a visual, what we're talking about here. First of all, you have your dorsal root of the spinal nerve, basically the little rootlets coming off of the spinal cord to form your dorsal root. Of course, immediately goes into the dorsal root ganglion at that particular point. And then below that, you can see your ventral rootlets that will form the ventral root. And then the dorsal and the ventral root will then merge into your spinal nerve. So your spinal nerve is the merger of the dorsal root and the ventral root. Then this is where the spinal nerve will split off into the two ramus, the rami. And so the dorsal, which will go toward the back, right? That'll be the dorsal ramus. That's the one that goes toward the back, and the trunk and the deep muscles of the trunk will kind of wrap around and head toward the back. And then the ventral ramus will kind of wrap around the front of you. And this is basically kind of wrapping around into the intercostals. So it's kind of like a barrel, essentially, that kind of wraps around the front side of your thoracic area. Now, this sympathetic chain ganglion system, we only have a little bit of it shown here, but what you see running lateral and parallel to the spinal cord is this series of ganglia that kind of form this parallel chain. This is called the sympathetic chain ganglion. And the communicating ramus basically will be connecting to these individual ganglia. So for instance, here's the dorsal kind of running to the back, and the ventral ramus will kind of give off an offshoot, which will be the communicating ramus that will go to the sympathetic chain ganglion. Now, the chain ganglia is something that's kind of like a series of sort of processing stations, substations, if you will, that will allow you to have extra processing specifically for the sympathetic division of your nervous system. So I want to kind of go through these different regions quickly. I don't want you to get too lost in all of the wires. There are a lot of them. I just want to kind of point out a couple of overall ideas and some kind of major patterns that we see here. So the first stop we're going to make is going to be the cervical plexus. So the cervical plexus, which is going from C1 to C4, this is basically going to be looking at your neck, the skin of your neck, and the posterior portion, the back side of your head. And so you can see, first of all, that from the ventral ramus, you'll actually see the roots coming out. That's in red. And then you'll see the branches in yellow as they start to split and merge and split and merge. That's basically the yellow portions. And then the green is other nerves that kind of pass through that aren't necessarily associated with it. Like, for instance, up here you have the hypoglossal nerve, and then the accessory nerves. So some of the cranial nerves that are kind of moving through here that aren't necessarily associated with the actual cervical plexus itself. Now, within the cervical plexus, there are a couple of interesting structures and one nerve that's worth knowing. The first interesting structure is what's called the ansa cervicalis. It's a loop that basically connects between C1 and C3. So basically, you'll see the ansa cervicalis here. And so you'll kind of start it at C1. So that's the branch off of C1. And it'll basically rotate around, and then it'll reconnect to C3. So that's basically your ansa cervicalis. And so what this is doing basically is looking at some of those infra or below the hyoid muscles. So that's basically what's innervating. Another important one that's worth knowing is this guy down here. So the phrenic nerve is definitely one that's worth knowing, at least knowing that it's in the cervical plexus structure. And so the phrenic nerve will basically come off of a portion of C3, and a C4 will merge together, and they'll bring in a piece from C5, and that'll basically be the phrenic nerve. The important reason the phrenic nerve is important is because this is the one that innervates the diaphragm. And so this is the nerve that you actually have to communicate with when you want to take a breath. And so this is a nerve worth knowing. This is also the nerve that gets irritated when you have the hiccups. Okay, so now let's go ahead and take a look at the brachial plexus. We'll just kind of move our way down. So we stopped off at C4, and now we're going to be taking a look at from C5 to T1. So ultimately, these are 5 ventral rami. They'll form 3 different trunks, and then they'll kind of move into divisions and cords and stuff like that. And so some of the branches that it gives rise to are like the axillary branch, the radial, the musculocutaneous, the ulnar, and the median. By the way, these should probably sound familiar, right? Because these were all the ones we were looking at in the cat. So in the cat, we were definitely kind of emphasizing that brachial plexus group. So there are smaller nerves, however, these are the main ones. The pectoral, long thoracic, the subscapular, which we saw. So these are smaller nerves, but these are also part of the group. So this is kind of what it looks like when you take a look at the brachial plexus. You'll see this looks kind of familiar. So you have that axillary artery, which typically is a very large artery that kind of goes through. And now we didn't really get a chance to see the artery. That's a major landmark in this area. But you can see that as you kind of follow that through, you can kind of see how it sort of merges and sort of divides and things of that nature. So when you take a look at the brachial plexus from C5 to T1, so you can see that you have the major spinal nerves coming off, and then they'll split into trunks. So for instance, C5 and C6 will form an upper trunk. C7 will form a middle trunk. C8 will form a lower trunk. And then from here, you're going to start splitting off. And so you can see some of the designations. So in red are the roots, the main roots that we're looking at, or the ventral rami. The trunks are the light blue. The purple are the anterior divisions. So those are the ones going to the front of the body. So here you can see you have a purple division that comes off of the first trunk. And that basically will kind of give rise to several nerves downstream. And the posterior divisions, the ones that are going more toward the back, are going to be in green. And then the split off into cords and then the major branches are in yellow. So you can see how you kind of basically subdividing, and then subdividing. And so ultimately, when you take a look at, say, for instance, the axillary nerve, the radial nerve, the musculocutaneous, the median, and the ulnar, these probably are nerves that are worth knowing. And it's going to help out because these are also the ones that tend to overlap with what we're doing in anatomy and the anatomy portion in the lab as well. So these are all basically going to the arm. That's why it's called the brachial plexus. And this is going to be controlling and innervating the upper arm. So here's kind of what it looks like as it starts to head into the upper arm. So you have the axillary nerve, which is going to be one that's worth knowing. So this one is basically coming from C5 and C6. So if you kind of take a look at the axillary nerve, so C5 and C6 are basically joining in the trunk, and then they'll kind of form a posterior portion ultimately that will merge and become the axillary. So this is kind of just showing you where that original source was. And so the axillary is important because what it's going to be working on is motion of an important piece, the teres minor, right, which is part of your rotator cuff. So that's your shoulder movement and also your deltoid, right? So this is basically what it's doing. And then the sensory is going to be cutaneous, that is your skin. It's going to be doing sensation of the skin as well. So you can see you have basically different cords that will branch into the different muscles. For instance, the teres minor is going from the posterior cord, the deltoid, and so forth. And the lateral and the medial cords will basically be going to the lateral and the medial side of the upper arm. So you're basically looking at this region up here, your shoulder region. Now, when you take a look at the radial nerve, also another one that's worth knowing, this one's going to be a cord, or coming from a cord between C5 and 11. So there's several different things here. And so the important thing is, as the posterior cord comes through, what's going to happen, now the posterior cord will move into the teres minor. So we're not looking at that particular part right now. So now we're looking at this next piece. So if you take a look at this, we were first of all looking at these upper portions here as it relates to the axillary nerve. Now what we're going to do is we're going to follow this little tip right here. And that little tip then becomes the radial nerve, which then starts to move down in the middle of the arm, the upper arm. So you can see it's innervating the triceps, the brachioradialis, a lot of the extensors of the lower arm. So you can see this is going to be innervating a lot of those extensors, and then it's going to be moving down on the radial side of the lower arm. And so that's why it's called the radial nerve. So the key thing here is it's really innervating a lot of these extensors, and you can see that here, right? So basically the extensor, 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 extensor. So a lot of these extensors are being fed by the radial nerve. Now, if you take a look then at the musculocutaneous, now you're looking at the lateral cord, this first portion of the lateral cord. So this one's basically going to come down. So this one up here was the axillary. This one was the radial. Now you're looking at musculocutaneous. This one is going to be going after a couple of others. So for instance, going to be innervating the biceps brachii and the brachialis. And then it's going to be moving down more the radial side. So kind of following along with the radial side, only in this case, it's doing a little bit less innervation. Most of its innervation is going to be in that upper forearm area. The forearm area, the radial side of the forearm area. So a lot of this is going after the flexors, right? So basically you have flexion of the shoulder, you have flexion of the elbow. And so a lot of these are looking at some of the flexors that are associated, in this case, with the upper arm. But this guy's originating from C5 to C7. The ulnar nerve, another good one. So this is coming from the lower portion of the brachial plexus. So you're looking at C8 to T1. In this particular case, now you're looking at the lower portion of the medial cord. And so it's going to be coming down the medial side of the arm and moving down into essentially mostly, if you take a look at it, mostly it's going to be looking at the flexors of the forearm. So for instance, flexion, flexion, flexion. And so a lot of the flexors are going to be fed by the ulnar nerve. It basically passes by the ulna, why it's called the ulnar nerve. And then this will kind of move down that medial side of the ulnar side of your arm. And so that was an easy nerve to find in the cat. We found that one pretty easily. And then the median nerve is coming off of the medial cord, but it's coming off of the extension that's just above the ulnar. And so the median is going to be moving down the upper arm, and it's going to also feed even more of the flexors of the lower forearm. So basically all those flexors that we learned about, palmaris longus, flexor carpi radialis, right, flexor digitorum profundus, and things of that nature. So the majority of the flexors of the lower arm are going to be fed by the median. And then that is, so you have ulnar coming down the ulnar side, radial musculocutaneous coming down the radial side, and the median kind of coming down right in the middle. Now, the important thing is, what happens is the flexor retinaculum, which is like a little wristband of connective tissue, will basically bundle all of these nerves that are coming from all these different areas. So you have the medial nerve, the radial nerve, the ulnar nerve, and the musculocutaneous nerve. They're all kind of coming down into this wrist area. And your flexor retinaculum is this little piece of kind of a wristband-like piece of connective tissue that kind of bundles all these nerves up. And the carpal tunnel is deep. So it's like a little pathway that a lot of these nerves will pass through. And so if you have carpal tunnel syndrome, then what you have is an irritation of the nerves that are passing through. It's usually the median nerve that's passing through that little carpal tunnel. And so you have like little passageways for these nerves to pass through so that they're protected and sheathed and not just exposed, because your wrist is a very exposed area. It's highly mobile. There's going to be a lot of strain and stress, physical strain and stress associated with that particular area. So this is a way to protect it. So a couple of other nerves, the smaller ones that we've been talking about, things like the pectoral, the thoracic, the subscapular, and so forth. So these are basically named after the regions that they're going after, right? So ultimately, the pectoral will go after the pectoral region. The subscapular will be going after the subscapularis, and the suprascapular will be going above the scapula. So it's kind of naming exactly where these different nerves are moving toward. Then as we start to move further down, we can take a look at the lumbar plexus. So the lumbar plexus basically is going to be associated with the ventral rami of L1 to L4, and the sacral is L4 to S4. Usually we kind of lump these together because the lumbar and the sacral oftentimes tend to work together in terms of their nerves. like for instance, when well, on Tuesday we managed to find the obturator nerve in the cat and also the femoral nerve in the cat. But if you recall where that was, it was more in the lumbar region, even though the nerve went down into the sacral region and then innervated the upper leg. So they tend to sort of work together because they're oftentimes in the same area and they're innervating similar structures. For instance, the major nerves that are associated with this area that are moving from this area, the lumbar region and the sacral region down into the lower limb, are, for instance, the obturator, the femoral, the tibial, and the common fibular. If you're keeping track, these should sound familiar to you because these are the ones that we were actually looking for and finding, for the most part, in the cats on Tuesday, for which I think we have some video footage, I do believe. I don't know, Sarah can probably confirm that. But
yeah, sorry, I was just trying to find the unmute button.
So that should be there because I know there was a couple of videos and there will be more. So we'll take more. It's not the last time you're going to have a chance to see it. So this coming Tuesday, we'll take a look at that. And I'll kind of do a little bit of a rundown on the cat to kind of show everybody what we saw. So there's that.
And I can post the video and the photos up tonight under the discussion area.
Good. So that should be there in case you missed it. Because I know all that kind of happened right there toward the end of lab. So the lumbar and the sacral plexus, you can see the same designation. You see the roots, the ventral ramus roots in red, and then the posterior divisions in green, the anterior in purple, and then the nerves themselves in the yellow. And so when you take a look at this, you can see that basically there's a lot of wiring. So they split and they merge and they split and they merge. And so a couple of things you want to make sure you make a highlight of, certainly the femoral and the obturator, and then the tibial and the common fibular or the peroneal nerves. And so these are going to be important pieces that you want to make sure that now there's a lot going on here, right? So this is basically the area where you start getting to the cauda equina, right? Where that horse's tail, just a big gamish of nerves and rootlets and things of that nature. So we want to kind of make sure we can keep it digestible. When you take a look at an actual dissection, you can actually see, now this is obviously really nicely and well done, but typically there's in the back is going to be a sheet of muscle. And so when we did the cats, a couple of landmarks that we found, first of all, was the iliac split. So where the abdominal aorta, which is right here, splits into external iliac arteries. And they go down the legs. So those become the femoral arteries. So smashed between that and one of the back abdominal wall muscles where you can see that nice shiny sheet. You actually saw this in the cat of tendon is going to be the obturator. That's this little guy right here. And we saw that in the cat itself. The femoral nerve then splits off from the obturator. We saw that in the cat. It's not so well seen here, however. And you can see a bunch of others. Now, if we really were getting more into a sort of a nervous focus class, we could easily dig into this a lot deeper, but that's kind of just an idea of kind of what it looks like and sort of what we saw. And you'll see that on some of the video footage as well. So when you take a look at your chords, your L2, L3, and L4 chords, we're going to start off first of all with the obturator. So the obturator basically is going to branch off of 4, and then it's going to be innervating and kind of going down through your hip, and it's going to be moving into these major medial adductors, right? So remember all those adductor muscles we were learning about, like adductor magnus and longus and brevis and gracilis, and all those adductors that are on the inside of your thigh are going to be innervated by the obturator. And so the obturator is going to be an important one to know, right? So this is adductors mostly of the thigh. Now, the femoral is going to basically be moving off of a kind of a branch. It's a merger basically of a combination of L2, L3, and L4. So here's the L2 piece that comes in, the L3 piece that comes in, and then finally the L4 that comes in and merges, and that's going to give rise to the femoral nerve, which is also moving into the medial side of the thigh. But in this case, what's happening is it's going to be going after the quad group, right? So that's going to be a really big component. So it's going to be going after the vastus lateralis, vastus intermedius, vastus femoris, rectus femoris, and things of that nature. So it's going to be going after your quad group, and that's going to be what's getting after. It's also going to be innervating a couple of your upper hip flexors, right, like psoas major and iliacus and sartorius. So it's going to be going after some of those upper leg flexors as well. So the tibial and the common fibular, the peroneal nerves, these together are typically what we refer to as the sciatic nerve. So a lot of times you probably have heard about the sciatic nerve. You know, people argue they have sciatica. It's kind of a shooting pain that kind of goes down the leg itself, and it creates problems. It can be painful. Nerve issues can range in their presentation. They can either be painful, or a lot of times what you'll see in kind of a nerve dysfunction is not so much pain as much as a kind of an odd sensation of like weakness of the muscle, and it can range a little bit, and so they're kind of bound together in the same sheath. That is just like kind of the way that the carpal tunnel is in the wrist. This kind of sheath kind of bundles together these two nerves into the sciatic, and so here is the combination of the two. So you have the tibial nerve, and the tibial nerve basically is going to be coming off of a combination of L4 and then all the way to S3. So it's going to be L4, L5, S1. So you can see L4 comes in, and then S5 adds its piece, and then S1, and L5, and then S2 adds its piece, and then S3 adds its piece, and then you start to get this big conglomeration that comes out as the tibial nerve. This is pretty typical nerve types of structure. So this is then going to be coming out, and it's going to be attaching to the back side of the leg. So all your flexors, right? So your hamstring group is going to be innervated by this nerve, as well as your adductor magnus. So your adductor magnus is going to have two innervations, the obturator and the tibial nerve. And then it'll go down the back side where it'll interact and innervate the gastrocnemius and the soleus. And it'll go all the way down the backside of the tibia, where it'll basically innervate the flexor digitorum longus, the flexor hallucis longus, and the tibial posterior. So this is a really important one. This is a major, major nerve. It's really hitting on most of the leg. In one way or another. Well, one way or another. So between the tibial and the obturator and the femoral, these are really, really major nerves that are designed to pretty much give all sensation and muscular control to your lower leg, upper and lower leg. So the medial lateral plantar nerves and the sural nerves, which are lesser in nature, but these are branches of the tibial nerve as it goes further down. So what about the fibular nerve then? So the fibular nerve, or the peroneal nerve, will basically come from L4 to S2. So you'll see basically it's branching off of 4 is adding its piece, 5, and then S1, and then S2. And this will kind of come down. It's going to be moving more toward the medial part of the leg. So this is your anterior view, the front as opposed to the back. So the tibial is going the backside or the posterior view. So that's the backside and the hamstring side. And this one's going the front side. So it doesn't necessarily have to innervate the quads because we've already got that covered. So it's going to be moving down, and it's going to be picking up a few of the smaller muscles, mostly the extensors, not always. But for instance, tibialis anterior, that's your shin muscle, extensor digitorum longus, extensor hallucis longus, so the extensor of your big toe, the fibularis or the peroneus longus and brevis. So these are some that we had actually on our lab exam. And so these are going to be innervated. These are like the extensors on the front side of the anterior side of the leg. So in general, you could actually think about these as sort of innervating the front side of the leg, whereas the tibial is the back side of the leg. And the loose designation is also true for things like the obturator and the femoral and so forth. So the deep and the superficial are basically branches of the peroneal nerve as they kind of branch off. So some of the other types of nerves that are coming off of this other area that are important. So the gluteal nerves, right? Obviously, those go to the gluteal muscles. The pudendal nerve, the ilioinguinal, the inguinal canal that's associated with your ilium and your the inguinal area where it basically kind of moves into the body. There's femoral, the cutaneous, that is the skin sensation of the femoral region, the genital region of the femoral area, so the kind of combination of the two. So you have different types of nerve basically kind of shooting out everywhere in the pelvic area and the upper femoral area. Now there's one left, the coccygeal nerve, or the coccygeal plexus, S5. And so ultimately, this is going to be looking at muscles of the pelvic floor. Now, if you recall, we when we got to the pelvic floor area, we saw a bunch of muscles there. We had that urogenital triangle, and we kind of moved quickly through that area. But all those muscles that are in that pelvic floor are being innervated by this coccygeal nerve, including the muscles of the anal sphincter that are tethered and tied to the coccyx itself. And of course the skin that's over the coccyx area. And so this is basically a couple of the different disorders that we have. I don't want to spend a ton of time on the disorders because you could probably spend a good amount of time on each one of these. But these are basically different types of spinal cord disorders, ranging from structural issues like narrowing of the spinal canal or inflammation of the area, so different types of inflammational types of disorders, some autoimmune conditions like multiple sclerosis, which will cause the breakdown of myelin, causing dysregulation. Some things can be caused by neurotoxins like tetanus, for instance. And so some are virally induced, like rabies, for instance. And these are the different types and there's more, right? There's more. So it just kind of keeps on going, like sciatica we mentioned before, right? That was just sciatic pain. And neuralgia, which is nerve pain. Leprosy is an interesting one. We don't really run into that one very often, but this is a bacterial disease. It used to be very common back in the old days in antiquity. And this is basically bacteria that kills the skin and also the peripheral nervous system cells. And so this used to cause a lot of problems. Polio is a viral infection of the central nervous system. Diabetic neuropathy, basically, ultimately you have decreased blood flow because of these high sugar levels that you have in a diabetic. And ultimately that decreased blood flow can create difficulties, including numbness in the extremities and things of that nature. And so you kind of get this sort of general numbing, extreme numbing. And that could be a problem that gets worse and worse. And myasthenia gravis is an autoimmune disorder that can cause major muscle weakness. and paralysis. Of course, neurofibromatosis is basically we have neurofibromas, basically benign tumors that are associated with your peripheral nervous system, and this is a genetic disorder. So you can see how a lot of these disorders, essentially anything that compromises the structure of everything we've talked about in terms of the nervous system and the structure of the nervous system, the structure of the neurons can create a neural disorder. And the nervous disorders are problematic because typically, if you do damage to your nervous system, well, once the tissue's gone, it's kind of gone, right? So nervous tissue is not very high on healing. So it doesn't divide, it doesn't replace. It's kind of gone. Once it's gone, it's kind of gone. Okay, that's the end of 12. But now, we're going to move into chapter 14, which is going to be integration. And then the last chapter we're going to have is 16, which is autonomic nervous system. So now you should be able to see the PowerPoint slides. And yes, I do know I've got some posting to do. Okay. So before we fire up chapter 14, how's everybody doing? By a show of thumbs. Okay. Good. Let's go into chapter 14. So between 14 and 16, we're really talking about some of the things that are going to be kind of pulling together a lot of our core systems. And we're going to be looking at how sort of the nervous system sort of pulls a lot of our core systems together. So we're going to be talking a lot about the communication of the nervous system between sensory information coming from other areas of the body and then control of the effectors. That's the outgoing information from the CNS. So it all starts, first of all, by collecting information, sensation, right? So ultimately, out in your body, you're going to have an array of sensory receptors, and these are all designed to basically bring information in your central nervous system so that you can sort through all that information. And so what this basically gives you is a sense of perception, which is your awareness of the sensation, so you can be aware of those senses. And so all a sense is basically is essentially the way the brain is collecting information and intelligence about the environment. That's essentially the world around you, and also what's going on inside your body. And so you have senses basically broken up into two halves. You have what's called general senses and special senses. So first of all, the general senses are generally are usually just kind of like spread out over your entire body. And so all of these senses will generate action potentials in order to send information back to the brain. But in a general sense, it basically will fire off kind of an action potential that will go to the brain called the generator potential. So oftentimes what will happen is somatic information, basically about the body and its environment. For instance, a good example would be like touch and pressure, temperature, pain, and proprioception. Now, proprioception, the rest of these we know, right? So touch, we know, we grow up with that one. Pressure, we know, we grow up with that one. Temperature, certainly, and pain, of course. Oftentimes, what most students kind of have a hard time figuring out and understanding is proprioception. Now, proprioception is essentially kind of a more of an abstract type of a sense, but essentially, for lack of a better way to describe it, it's essentially your awareness of body position and movement. Now, it's not necessarily your conscious awareness of body movement and position, rather your body's awareness of movement. For instance, your body knows where your muscles are. How does it know? Well, proprioception is the sensory pathway that allows your body to know, for instance, where your brachioradialis is, right? So there's kind of a sense of your body knowing where everything is in your body, and that's kind of proprioception. So basically, the sense of body position and movement. Now, those are somatic in nature. There's also what's called visceral, right? So these are essentially associated with your internal organs, your visceral organs. And oftentimes these will bring back information associated with pain and pressure that's associated with those internal organs. So those are all general senses. It's kind of like a global information grab on a lot of different levels for a lot of different inputs. Then your special senses are typically what we would think of as our five senses, right? So for instance, our smell, taste, vision, hearing, balance, things like that. And so the reason why they're special is because we actually have to create a very special organ in order to pull off these senses. So with the general senses, we don't have to do that. Basically, we just put a receptor out there and we just collect the information. Whereas with the special senses, we have to create a little bit more structure so that we can actually have an entire organ whose job it is to receive this information. And so the receptors in these special senses will basically create what's referred to as a receptor potential. And in this case, they'll release a neurotransmitter that will basically bind to the receptor on the membrane, and then of the neuron, and then it will it travels to the brain. So you kind of have the neurotransmitter involvement a little bit in this particular situation as well. So here's how your senses are broken down in terms of a flow chart, right? So your senses are broken into general and special, and then the specials are going to be your dominant five senses, having to create special organs in order to pull those off. And the general senses are going to be broken into the somatics, which are basically in the skin, muscles, and the joints. So this is your touch, pressure, proprioception, temperature, and pain. And the viscerals are associated with your internal organs, which are pain and pressure. So this is basically for those of you who like flowcharts, and this is for those of you who like tables. Basically the same information for the most part, with a little bit of extra, right? So you can see your somatic group here in your general senses, and your visceral here in your somatic and your general senses. Now, the piece that we add to this are the types of receptors. For instance, for most of your somatics, for touch, for instance, you have what's called mechanoreceptors. Basically, they are physically distorted. And so that's where the compression of the receptor comes in. So a squeeze, for instance, or a manipulation of a physical distortion of the receptor is what creates and activates the receptor. And then over here, we have specific types of structures that will create this. For instance, the Meissner corpuscle in skin is the touch receptor. The hair follicle receptor is a stroking receptor. Texture is coming from the Merkel disc, again, in the skin. The Pacinian corpuscle is also in the skin. The Ruffini end organ is a kind of a stretch organ. You have free nerve endings in the skin. Again, Merkel discs or the Merkel structure. And so you can see there's different types of structures here. Stretch, we just talked about the muscle spindle, right? That's a stretch organ. Same thing with the Golgi tendon organ. We just talked about that one, right? That's for proprioception. And so you have different types of structures that will help you to understand the nature of what's going on in your body. So those are all different types of receptors. Notice temperature has its own receptor, a thermoreceptor, and pain has its own receptor, a nociceptor. So ultimately, with pain, what you do is you actually irritate the nerve endings with whatever's causing the pain, either mechanical insult, chemical exposure, or thermal exposure can create the pain. Same thing's also true for your visceral. It's just that you're working with the organs. Visceral is going to be associated with nociceptors, right? That's going to be irritation of those nerve endings in your organs. And pressure is going to be mechanoreceptors associated with compression or distortion of those visceral organs. And then your special senses have very specific types of receptors. For instance, for smell and taste, you've got chemoreceptors that will basically be initiated once a molecule is bound to membrane receptors. For instance, if a smell molecule binds to the membrane receptors, then it'll initiate the response. Same thing with taste receptors binding to your taste buds. Photoreceptors are found in the vision area. Your rods and cones are photoreceptors. These are light sensitive. That's how you initiate the response. And then hearing and balance are both mechanoreceptors. So ultimately, you bend little microvilli on receptor cells, and that's a physical distortion in a structure called the hair cells that will allow you to be able to initiate the response. So a mechanoreceptor is basically going to be specialized for physical distortion of one site or another. So either by compression, by bending, by stretching. And so this will basically lead to the ability to sense touch, pressure, proprioception, and hearing and balance. These are all driven by mechanoreceptors. Chemoreceptors are basically the chemical attaching to a receptor and then initiating some sort of a signal. For instance, in smell and taste, thermoreceptors are going to be temperature sensitive. So basically they will respond to temperature changes. Photoreceptors will respond to light. And nociceptors are going to be either mechanical, chemical, or thermal, but it's always going to be irritation and therefore pain. So sensory receptors, which are based on location. So these are based on what they detect. These are based on where they are. You have cutaneous receptors, which obviously would be the ones in the skin, and you have a lot of different receptors in the skin itself. You have visceral receptors, which are associated with your organs, and then you have proprioceptors, which are essentially usually joints, tendons, and these oftentimes will be involved in stretch detection. And then, of course, the structure of some of these 8 types, you can see are going to be some of the lists that we saw on the table, right? So for instance, you have free nerve endings, that's going to be one of them. The Merkel disc, which we see in skin, the hair follicle receptor, which is also in skin, the Pacinian corpuscle, which is deep pressure in the skin, the Meissner corpuscle, which is light touch, that's also in the skin, the Ruffini end organ, right? That's number 6. The muscle spindle, which we just got done talking about, that's number 7, and the Golgi tendon organ, which is number 8. So these are the 8 different types of sensory receptor structures that we deploy throughout the body. And so these are just basically this is like the table structure for those of you who like tables. So let's go ahead and take a look at some of the general sensory receptors. Now, since we didn't cover the skin chapter, this may look a little bit foreign, but this is basically would be review from the skin chapter had we covered it. So what I want to do is I want to basically kind of cover what we're looking at here. So you have a little bit of an intro on this. We're looking at a skin section, and the skin is going to be made up of 3 different areas. the upper layer of the skin is called the epidermis. So there's 6 layers here of keratinocytes, basically skin cells, flat squamous skin cells that it's stratified. And so this forms the upper portion, the portion that you basically put lotion on and things like that. So this is going to be the epidermis. The lower portion of that epidermis is the mitotic layer, and that's constantly making new skin cells to replace these skin cells that you slough off. Now, immediately below that is the dermis. This is basically a connective tissue layer that has 2 regions, the papillary layer and the reticular layer. Now, underneath the epidermis are what's called these dermal papilla, which is the reason why it's called the papillary layer. It's basically an area where the connective tissue of the dermis pushes up into the epidermis and creates these little ridges. That's what creates the ridges of your fingerprints. And so within those little pockets or those dermal papilla, you're going to find one of the sensory structures, the Meissner corpuscle, which is basically involved in touch, light touch, 2-point discrimination, your ability to be able to feel the difference between a point, say, for instance, here, as opposed to here. That's the Meissner corpuscles allowing you to do that. So you can see how they have connections, little innervation connections that are basically sticking out and attaching to the epidermis. And then they're collecting that information and then moving that in toward the nervous system. Now, deeper down into the dermis, which is the reticular layer, you're going to see another structure called the Ruffini end organ. And so this one, its job is to detect continuous touch or pressure or depression of the skin. So it kind of like gives you more long-lasting sensation. Now, deeper still in the dermis, which is this purple area, is going to be another sensory structure called the Pacinian corpuscle. It basically has this sort of lamellated structure. It's like little layers around it. And this is going to be that deep cutaneous pressure, vibration, and proprioception. So it allows your body to know essentially spatially where everything is. And so if you feel pressure but not pain, then that's the Pacinian that's telling you that. Around your hair follicle, you have little nerve roots basically wrapped around it. And so this will basically allow you to sense light touch. Like, for instance, when the wind blows through your hair and you can feel that your hair is moving, that's the hair follicle receptor. Or if I were to come up behind you and kind of blow in your hair, you wouldn't know I'm there necessarily, but you would feel your hair moving and knew something was moving. We've all done this before, right? We've been outside and a bug flies into our hair. We didn't see the bug, but we can feel it because we can feel it moving our hair around. Those are the hair follicle receptors. Anybody ever experienced that one? The old bug in the hair trick? Happens to me just about every year when I go out into the yard and work on the sand cherry. Usually the bug is a Japanese beetle, but usually you can feel it crawling around in there. Now, the Merkel discs are going to be up further, locally, kind of throughout. There's going to be a good population of them in the epidermis, and you can see them spread out as well along the hair shaft and the hair root, which is down here. Touch detectors and very superficial pressure. So if I gently touch the surface of your skin, your Merkel discs, and also your Meissner corpuscles will allow you to be able to sense that light touch. And very, very gentle pressure. If I start putting a little pressure on there, you can feel the pressure also through these Merkel discs. And then also in this epidermal region, you have nerve endings, which oftentimes will be responding to painful stimuli, temperature, and things. These will be responsible for some of those sensations. Now, the lowest portion of your skin, which is your subcutaneous tissue, which is mostly fat, you'll have blood vessels and nerves in there. They're typically not a lot going on there, except for just conduits, basically passage of nerves going off to the spinal cord itself. So, what's the moral of this story? The moral of the story is that there's an awful lot of general sensation that's basically wrapped into the structure of your skin. So, your skin is actually a very complex and a very multifunctional organ that does more than just cover you like a Christmas present wrapping, right? It basically has a lot of functionality in it. And this is just focusing in on the nervous functionality. So, this is where you set out like a full array of different types of nervous receptors to collect all kinds of different information from your outside world. Okay. So, the free nerve endings. First of all, before I continue, do you guys have any questions? Or are you guys good? Or rather, what I should probably ask is, are you guys numb? No pun intended. Sorry to hear that, Daniel. I know I'm one of them, aren't I? So I guess I'm adding to your misery, for which I apologize, but I guess that's occupational hazard, right? So let's take a look at some of these structures. Um, the nerve endings. These are the easiest ones, right? Because they're just basically offshoots of the nerves. And so, really easy. All you have to do is just basically shoot off the ends of the nerves. And this is a really, really common receptor because it's super cheap and easy to make, right? So, it's a very easy thing to do. You don't have to come up with some sort of crazy structure. And not only that, but you can collect a lot of information with just the nerve endings, right? So, for instance, pain, sensation, temperature, itch, and movement. And so you can spread these out pretty much anywhere you want in the epithelium, in the connective tissue. So you can pretty much put them wherever you want. And so this is going to be where your temperature sensation is coming from. For instance, we oftentimes refer to these as cold receptors, warm receptors. So we oftentimes have a lot more cold receptors out there than warm. And so we can sense a lot of cold, not so much warm. And then, of course, the pain will kick in when we start to get extreme cold or extreme heat. So you can cross over between just, wow, that's cold, to ouch, that hurts, it's so cold, or ouch, it hurts, it's so hot. So you can see how you can basically transform from temperature and you can move into pain. And so you can do that with either. Now, the Merkel, the tactile discs, are basically branches of an axon that basically innervate the epithelial cells and the bottom regions of the epidermis. And so these dermal papilla is basically where you're going to find some of these little discs. And so these oftentimes are those light touch and superficial pressure guys. The hair follicle, like we mentioned before, basically is wrapped in these receptors at the root. And so this will allow you to sense when your hair is bending in the breeze. Your lamellated corpuscles or your Pacinian basically takes a dendrite and wraps layers around it to create this kind of onion-like structure. And so it's typically very deep in the dermis, right next to the boundary between the dermis and the hypodermis. The hypodermis is the same as the subcutaneous region. They're synonyms. So it's right there at the boundary between deep dermis and hypodermis, which allows you to be able to sense deep pressure. That's the reason why that's what it does. It does that deep cutaneous pressure and vibration. So If you happen to have a Pacinian corpuscle in a joint, then it can be involved in that body positioning proprioception role as well. So the Meissners are basically going to be tucked just in that dermal papilla, right at the junction of the lower epidermis and the upper region of the dermis. And so when we said this is your 2-point discrimination. So if you were to basically take some calipers and basically put the sharp points at 2 different points, then you can tell that there's 2 different points there. And so that is your Meissner corpuscles that are able to tell you that. And so this is what those calipers are for in the lab. I think we should have some calipers. I think I forget if Sarah and I found them, but I know they're there somewhere. I think there's a bag in the back of calipers, but this is what this is for. It's basically kind of sensing your 2-point discrimination, and that's going to be the next lab. That's not something we did last Tuesday. That's going to be something that we're going to be doing in this lab coming up. Right, that's the last lab we're going to have. The Ruffini organ basically is mostly in the fingers. So this is like continuous touch and continuous pressure. You have a lot of nerves in your fingers. That is what allows you to be able to sort of feel your world, essentially. And it gives you a lot of fine sensation. And so this is the reason why a lot of times finger injuries are really painful, because you have lots of nerves and nervelets and nerve receptors out there, because that's one of the major ways that you sort of experience your world and you take in information from your world. Oftentimes it's not uncommon. We do it subconsciously. We don't even know we're doing it. We'll walk by something and we'll just touch it, right? We'll just touch it. Why? Because that's kind of the way we sort of receive information from the outside of our world. And of course, our spindles, we talked about this one already, right? So these guys are involved in kind of the stretch response associated with muscles, and also helps to coordinate contraction of muscles. It also helps to tone up our postural muscles, and it's part of that stretch reflex. We talked about that in the last chapter, so I don't want to go all the way back over that, but those spindle muscles are the same thing. The Golgi tendon organs, which we also talked about last time, is also, right, that's part of our reflex to keep from overexerting our tendon, so we don't want to snap our tendon. It's also proprioception associated with our tendons as well. So that positional information associated with our body. The responses of our sensory receptors, basically when our sensory receptors receive information, is what'll happen is when they receive a stimulus or an interaction, then it'll create a graded potential. And we talked a little bit about graded potential in chapter 11. And so in this case, the graded potential is called a receptor potential. Remember, that's for like especially for like a special sense. And then ultimately you have your receptors kind of broken into 2 different types. The primaries are the situation where the axons themselves conduct a potential, and it's responding to the receptor potential. So the primary action potential coming from the axon is a response to the receptor potential. So essentially, a lot of the receptors that we run into are of this type. A secondary one is a little bit different. This is basically one where the receptor potential causes a release of neurotransmitters in this case. So rather than like a direct kind of a conduction, you release transmitters that will then bind to receptors on a neuron, and that will cause the receptor potential. So rather than creating the receptor potential directly, which will then create the axon action potential, you'd use a neurotransmitter to create the receptor potential which will then kind of grow into that. So you kind of like a neurotransmitter or a molecule that creates the sense, right? So that's basically what we're talking about. So for instance, if you're looking at your special senses, smell, taste, hearing, and balance, you could imagine that some sort of a molecule, a smell molecule, will trigger the action potential, or some sort of a taste molecule will trigger the action potential. So that would be like a secondary receptor. So here's a comparison of the two. So the first one we're going to be taking a look at is the primary receptor, and then the second one we're going to take a look at is the secondary receptor. So in the primary situation, here we have an example we're going to use is a Pacinian corpuscle, right? That's that deep pressure corpuscle in your skin. So generally speaking, the stimulus will come in, and it's usually a type of mechanoreceptor. So that pressure will come in, it'll distort the corpuscle, and that pressure stimulus will then trigger the sensory receptor response by generating a graded potential. And that graded potential will be referred to as your receptor potential. So the physical distortion or the mechanical reception of the pressure will cause the Pacinian corpuscle to basically trigger this receptor potential. And then ultimately, when that graded potential reaches threshold, we all know what happens then. We learned about that in chapter 11. Then the action potentials will trigger, and then ultimately they'll start to move down the axon to the central nervous system. And so that's basically how you kind of ignite that primary receptor. Now, in a secondary receptor, in this particular case, you got something a little bit different. So for instance, in this case, you have some sort of a chemical stimulus, which will basically be received by a chemoreceptor. Like for instance, in this case, it's a taste cell. And so like let's say it's a salty kind of a substance. Then what's going to happen is once that chemical stimulus is received by the chemoreceptor, then the sensory receptor will basically create your graded potential. That's your receptor potential. So that's step 2. And then ultimately the sensory receptor, so that is to say that the receptor potential, instead of directly moving into action potential, will actually do something crazy. So what it'll do is it'll kind of put this other extra step in here, in which case the sensory receptor will release a neurotransmitter. And it'll basically move it into the synaptic cleft. Why? Because the taste cell is not connected to the neuron. So it's got to pass this on to the neuron itself. See, that's different with the primary. With the primary, the actual sensory receptor is connected to the neuron, to the axon. And as a result, when the sensory receptor creates a receptor potential, then it can automatically directly pass it on to the axon. But since in the secondary response or in the secondary receptor, the receptor is part of a cell that is not connected to the axon. And so in order to be able to relay what it has sensed, what the information that it has to bundle that up into a neurotransmitter that can then relay that information to the axon. And that's exactly what's happening here. So because they're not connected, then the receptor potential will cause the release of the neurotransmitter, which will then go across the synaptic cleft, and then it'll stimulate the axon. So it'll basically rekindle the action potential in the axon by igniting that action potential. It'll reach threshold. So that stimulation will reach threshold, and then that'll fire off an action potential, which will then move down the axon toward the CNS. So it's just basically how you have your receptors set up. Either your receptor will be directly tethered to the axon, in which case it can directly fire that axon off, or the receptor will be disconnected from the axon, in which case it has to communicate that to that axon through a neurotransmitter. So one of the important things, kind of interesting things that we see in kind of receptorology, if you will, is adaptation, where basically you have constant exposure to stimulus, but then what happens is you start to accumulate decreased sensitivity. And so this can adaptation or decreased sensitivity can happen either at the receptor locality. Essentially, the receptor is just saying, oh, well, we're not going to listen to that anymore because the stimulus is just so common. Or it can also be at the level of the brain. Your brain can also desensitize to certain stimuli. It can choose to basically ignore the stimuli that it wants to ignore. Your brain can oftentimes work in an additive way. It can fill in blanks basically, or in a subtractive way. It can ignore constant stimuli. And so that's something that's just kind of an interesting adaptation is something that happens actually across your nervous system. Now, proprioceptors, basically, when you take a look at this, the proprioceptors are taking in information and they're communicating not just position, but movement of your various body parts, communicating the weight of an object that's held in your hand, the range of movement of a joint. So notice this is all basically parameters, if you will, of the things that are happening in your joints in particular, usually. Right. So it's kind of like not just position, but movement, like your ability to sense that one object is heavy than another. That's proprioception. So that's kind of what the proprioception is kind of does a lot of sort of lot of things like that. Now, tonic receptors versus phasic receptors. These are two different ways that you can sort of categorize receptors. Basically, an tonic receptor, they basically move very slowly. They accommodate very slowly. For instance, in a Merkel disc or a Ruffini end organ, these guys sort of move very slowly, whereas opposed to phasic receptors are accommodating basically very rapidly, usually very much more sensitive to changes, quick changes, and things like that. So Pacinian and Meissner are examples of phasic receptors, essentially associated with the speed with which they are sensing some things. So we are just about at our witching hour, and this is actually a really good slide to stop at, because in order to cram this one in there, it's probably make your head spin. But this is one that we can start with, and we'll kind of start covering a little bit about how the different pathways are integrated into the spinal cord. And we'll start looking at some of these next time. We'll do that on Tuesday. So until then, study hard for the exam.